Okay, last time we began discussing non-Mendelian genetics. That is, genes that do not behave exactly the way Mendel thought all genes behave. Exceptions to Mendel's rules and laws of heredity. Specifically, last time we discussed incomplete dominance and codominance. And remember, our example for incomplete dominance was snapdragon flowers, where the heterozygous plants have pink flowers. They have a red allele and a white allele. Neither is dominant, and so the flower color becomes pink. And then for codominance, I used the example of blood type, how blood type is inherited. So we're going to continue today discussing non-Mendelian genetics. So before we leave codominance completely behind, another great example of codominance is in shorthorn cattle. There's something very similar in certain breeds of horses as well, but specifically here we're talking about the coat color, the fur color of the animal. And you can see in this particular species of cattle, there are three phenotypes. This sort of brownish color is actually referred to as red, and then obviously you have white, and then you kind of have what seems to be sort of an in-between color, but I'll talk in a moment about what is really going on with this animal here. So first of all, the cows that are quote-unquote red in color are genotypically RR. They have the genotype RR. That's two alleles that code for this reddish color fur. And the cows that are white are homozygous R'. So both alleles coding for a lack of pigment or white color hair. And so if you breed a red steer and a white cow, or vice versa, could be a white steer and a red cow, the resultant offspring, the heterozygous offspring, are going to be RR', and the color is described as roan, R-O-A-N, roan. It looks like it's an in-between color, but actually if you could get down at the skin level, you would see that basically every hair on this animal is either this color or this color. Each individual hair is either red or white. But because the red and white hairs are dispersed across the animal's body, when you step back from it, it kind of looks like this in-between kind of rusty red color, which is described as roan. So again, in shorthorn cattle, RR for red, R', R' for white, and then roan is the phenotype whenever we have a heterozygous animal, that is RR'. So with that in mind, a little sample problem here, actually 4 sample problems. Figure out the expected phenotype ratios for each of the following crosses. And you can see the first one is, that would be a roan steer bred with a roan cow. The second one is roan crossed with red, white crossed with white, and then finally white crossed with roan. So this would be a good time to pause the video, see if you can work out each of these 4 crosses, either by doing Punnett squares, or maybe you can just kind of picture it in your head and see if you can figure out what phenotypes we are expecting to see in the offspring. Go ahead and pause the video and give it a try. Well, let's see if you came up with the right answer. Roan crossed with roan, this would be analogous to a pink snapdragon crossed with a pink snapdragon. And you get exactly the same kind of result. You get 1/4 red offspring, 1/4 white offspring, but half of the offspring are expected to be roan. Roan crossed with red, half of the offspring are expected to be red, half of them are expected to be Roan. Notice if I cross white with white, that's R prime, R prime, crossed with R prime, R prime. So of course, all of the offspring have to be R prime, R prime, and therefore they all will be white. That's the only possible phenotype that could be produced. And then the last one, white, R prime, crossed with Roan, which is R prime. And you see that we would expect half of the offspring to be Roan and half to be white. Okay, and again, this is fur color in shorthorn cattle. And as I said, in certain breeds of horses, you see the same sort of pattern with red, white, and Roan as well. That's a great example of codominance. All right. I wanted to make this point that your textbook author made, and I thought this was a point worth bringing to your attention, that I think most people just assume that if we identify an allele as being a dominant allele, that automatically means it's more common, that dominant phenotypes happen more often and we're less likely to see recessive phenotypes. And in one sense, that kind of makes sense, because remember, the only way you can show a recessive trait, the only way we ever see the recessive phenotype, is if the individual has 2 copies of the recessive allele. You have to be homozygous recessive to show the recessive trait. So it seems like that should be rare. Well, the problem with that logic is calling an allele dominant or recessive says absolutely nothing about how common that particular allele is in the gene pool. So, for example, this is a really good way to illustrate this, and this is totally true. In humans, there's a condition called polydactyly, poly meaning many, dactyly meaning digits or fingers. Polydactyly is an inherited characteristic where a person is born having either 6 fingers on a hand or 6 toes on a foot. So it's an extra digit, or at least one extra digit. And that is actually caused by a dominant allele, which means the vast majority of people on Earth, like 99% of all the people on the Earth who have just 5 fingers on each hand and 5 toes on each foot, those people are homozygous recessive. So even though it's a dominant allele that causes you to have 6 fingers on a hand, it's an extremely rare allele that very, 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 very few people on the Earth have. And so this is consequently a very uncommon trait. So again, a dominant allele is not necessarily the one that's more common. Sometimes the recessive allele and the recessive phenotype are much more common than the dominant. So that's an important thing to understand. Okay, so we're going to continue now discussing exceptions to Mendel's rules, things that we know that sometimes happen with certain genes, but these are things that Mendel did not know about. And so the next one on the list is called Pleiotropy. Pleiotropy is when one gene has multiple phenotypic effects. So Mendel thought it was simple. He thought, okay, there's one gene that controls the color of the flowers, and there's a totally separate gene that, controls how tall the pea plant is, and then there's a gene that controls the color of the seeds, and then there's another gene that controls the shape of the seeds. Well, sometimes you have a single gene that has many effects, or at least more than one effect, and that's pleiotropy. So here's 3 or 4 examples that I'm going to give you here. PKU, which is an inherited disease in humans. It stands for a really long word, phenylketonuria. Only humans that are homozygous recessive have this trait, and it causes the person to be missing an enzyme And the job of that enzyme is to break down the amino acid phenylalanine, which comes into our body through the proteins that we eat. So these folks who have PKU, they're missing an enzyme. They cannot break down phenylalanine, and it builds up to dangerous levels in the bloodstream. This causes many, many changes and adverse effects on the brain, but it also affects the heart and the circulatory system. So it's one gene that affects more than one thing in the body. It has more than one phenotype that it has an impact on. Okay, a really common one that you'll see in biology textbooks in chickens is a trait called frizzled feather trait. And you can look this up if you Google frizzled feathered chickens. You'll see some interesting photos. It's what it sounds like. These are chickens that instead of having smooth feathers that lay down nice and neat, they just kind of look wild. They look disheveled, and their feathers are just sticking out every direction, and they just don't really look normal at all. So that's a single gene that causes the feathers of these chickens to be frizzled, or, you know, kind of frizzy looking. But that same gene also speeds up the metabolism of the chicken, so they tend to eat more and eat more often. And it causes limitations in their fertility, so they have a harder time reproducing. So it's one gene, but it affects the feathers, it affects the metabolism and how they process food, and it also affects how they reproduce. So one gene, multiple effects. Pleiotropy. Sickle cell anemia in humans is another inherited genetic disorder. People that are homozygous recessive for this gene have abnormal hemoglobin protein, which causes the red blood cells to be sickle-shaped, hence the name, sickle cell anemia. Instead of nice, round, smooth blood cells, they're shaped like little quarter moons, and they do not flow smoothly through the capillaries of the body, and they get stuck, and they clot, and they form very painful blood clots that restrict blood flow and affect stamina and the delivery of oxygen throughout the body. So that's the main effect that this gene has. But it also has another effect, especially for folks who are just heterozygous. They have one copy of this allele. Even though it may cause, if you're heterozygous and you have one sickle cell allele, you may have some abnormally shaped red blood cells, but you also are resistant to malaria. Malaria, as you know, is caused by mosquitoes. Mosquitoes carry a parasite. Some, not all mosquitoes, but some mosquitoes carry a parasite called Plasmodium, and the parasite is transmitted into the body by mosquito bite. And for many people, once this organism gets in the body, it reproduces and it causes really bad fever and can be deadly. But for folks who inherit one copy of this sickle cell allele, their blood cells become incompatible with the parasite. So it protects them from this horrible, deadly disease called malaria. So again, one gene that affects more than one trait. It affects the shape of the blood cells, but it also affects whether or not you are susceptible to malaria. And then a final example of pleiotropy, Marfan syndrome, which is actually an inherited dominant trait in humans, thankfully a very rare dominant allele. If you're heterozygous and you have one copy of this dominant allele, it causes the person to grow up to be unusually tall and thin with long fingers and long arms. So it affects the stature, it affects the shape of the body, but it also affects the way the aorta connects to the heart. So this is the largest artery that comes off of the heart to carry blood throughout the body. It causes the aorta to have an abnormal bulge which is subject to rupturing. Basically, it increases the chance of an aortic aneurysm, which is, of course, very deadly. So it's one gene that affects the shape of the body, and it affects this blood vessel that's connected to the heart. One gene, more than one effect, is called pleiotropy. Okay, next on our list of exceptions to Mendel's rules is a phenomenon known as epistasis. Epistasis is when the phenotypic expression of a gene alters the phenotypic expression of another gene. So basically, it's when one gene has the ability to override a totally separate gene. And one of the great examples of this is the way fur color is determined in rodents like rats. But who in the world wants to learn about rats? So I've got a much more adorable example to illustrate how epistasis works. We're going to talk about Labrador retrievers, which is my favorite breed of dog, and they're beloved by many. And if you know much about labs, you know that now breeders have tinkered with some of these colors over the years, and so there are some new varieties now, but the standard classic 3 colors that you see in full-blooded Labrador retrievers are black, brown, which we call chocolate, and yellow. Black, chocolate, and yellow. And so we're going to learn how coat color is inherited in Labrador retrievers. And this will be a great example of epistasis. Now remember, this is going to be one gene that alters the effect of another gene. So there's 2 separate genes. The first one is called gene B. And this is the gene that codes for the color of pigment, which is either black or brown. So big B is the dominant allele that codes for black fur in labs. And little b is the recessive allele that codes for brown, which again is referred to as chocolate. Now, that means homozygous dominant is going to have black fur. Heterozygous is still going to have black fur, indistinguishable from each other. And then the only way to have a chocolate lab is little b, little b. So what about the yellow labs? Well, remember there's a totally separate gene called gene E. And I'm not sure why they chose E. I'm not sure what the E actually stands for. It's kind of a weird choice, but this second gene, gene E, codes for whether or not the pigment, which remember can either be black or brown, whether that pigment is actually deposited into the hair follicle as the hair grows. So if the pigment is not deposited, it doesn't matter what color it is. You're not ever going to see that color unless it's deposited into the hair. So any Labrador retriever hair that has no pigment deposited into it is going to be kind of this default blondish color, which we have come to refer to as yellow, kind of a blondish color. All right, so you get the idea. So big E, we're going to see some color, either black or brown, in the hair. Big E, little e, heterozygous, same thing. You're still going to see some color. It'll either be black or brown, depending on what the other gene says. But the only way you get a yellow lab is if the dog is homozygous recessive for the pigment deposition gene. That is little e, little e. That means no pigment is deposited in the hair, so it doesn't matter if it's black pigment or brown. It's never going to go into the hair, and so the hair is just going to be the basic yellow color. Okay, so let's look at this Punnett square. I think this illustrates pretty well for you what's going on with epistasis and Labrador retrievers. So we have 2 black labs that are being bred together. Notice they're not only both black labs, but they're heterozygous. Both of them are heterozygous for both genes. So heterozygous black big B, little b, both of them. And then they're also both heterozygous for the pigment deposition gene. That's the E gene. And so when you cross the two labs, notice it's a dihybrid cross, right? Because we're looking at two separate genes. And so it's a 16-box Punnett square. If you look at all the possible offspring that could be produced from these two parents, any pup that gets at least one big B, and at least one big E will have black fur. So big B, big B, big E. Big E is black fur, but so is big B, little b, big E, little e. That's also black fur, because all it takes is one big B to code for the black pigment, and all it takes is one big E to get that pigment into the hair. So a chocolate lab is going to always be homozygous recessive for the first gene, little b, but it would also have to have at least one big E. Okay, let me get my laser pointer going here. So yeah, so homozygous recessive for the first gene, but at least one big E in order to get that brown color into the hair. And then notice any pup that is little e is yellow. And it doesn't matter what the other gene says. It doesn't matter that this gene is coding for black. It doesn't matter that these two little b's are coding for chocolate. The little e overrides the pigment color gene. Remember, that's what epistasis is. It's where one gene can be expressed in such a way that it affects how the other gene is expressed. And that is called epistasis. Okay, so let's see if you understand how epistasis works. Here is a sample problem. It says use a dihybrid Punnett square to show the following cross. So here are the two lab parents. First of all, what color are these two parents? So this one is black. It's heterozygous black. And notice it's heterozygous for both genes. So this would be a black lab. And it is crossed with a yellow lab. Remember, little e means that it doesn't matter what this gene says. That this is actually coding for black pigment, but this is a yellow lab because it's little e. So this is your dihybrid cross. Go ahead and show me the cross between these two parents. See if you can figure out what proportion of the puppies are expected to be black, chocolate, and yellow. Pause the video, work it out, and then you can unpause and check your answer. Okay, so here's my Punnett square. This was the heterozygous black lab parent. And so these are the four possible gametes that could be produced by that black lab. And then here is the yellow lab parent. And notice it can only make two different types of gametes. It's either going to be big B, little e, or little b, little e. Those are the only two possibilities. So even though this is a Punnett square, I didn't fill in all 16 boxes. I could have, but that really would have been a waste of time. I can see all of the possible combinations of sperm and eggs just with an 8-box Punnett square. And so here is the solution. These are the 8 possibilities. And now it's just a matter of counting how many of these potential puppies are expected to have black fur or yellow or chocolate. And so I counted them up, and out of 8, I identified 1, 2, 3 that would have black fur. So that's a 3 out of 8 proportion. And then I identified only one that would be chocolate. Remember, it's got to be little b to have the brown fur, but it also has to have at least one big E, and there was only one like that. And then the other half of the litter I would expect to be yellow labs. So we should have quite a few yellow pups in this particular litter, approximately half. All right, so that is epistasis. Okay, the next violation of Mendel's rules is something known as polygenic inheritance. This is when 2 or more, sometimes it's 3 or 4 or 5 genes, affect a single phenotype. So this is really the exact opposite of pleiotropy. Because remember, pleiotropy, which we talked about a few minutes ago, was a single gene that has many different phenotypes that it affects. It's a single gene that affects many different things. Well, this is the opposite. This is many genes, or at least 2, that affect one trait. Okay? Usually with polygenic traits, there's a very broad spectrum of phenotypes. So it's not like pea plants where it's either purple flowers or white. Or it's not even like snapdragons where it's, either red flowers, white flowers, or pink. Usually with polygenic, there's a very broad spectrum where you might have, 20 different shades of color possible, or 20 different sizes or shapes, or whatever the trait is. And so some examples, and there are many, many, many human examples. In fact, I think it would be safe to say that the majority of human inherited traits are polygenic. Certainly things like height. So far, at least 180 different genes have been identified that have some impact on how tall you will eventually grow up to be. That's a lot of genes. That's definitely polygenic. It's not like pea plants where it's, big T for tall and little t for short. It's way more complicated than that. At least 180 different genes that affect height. Human eye color is affected by at least 3 different genes. Human skin pigmentation, how darkly pigmented human skin is probably affected by as many as 6 or 7 different genes, and maybe way more than that, but certainly it's a number of different genes. We're going to look at a highly simplified example of human skin pigmentation, and we're going to assume that it's only affected by 3 genes, but just know it's really more than that. It's really more like 6 or 7 or 8 probably. With human skin pigmentation, there are many separate genes that affect how much melanin, that's the human skin pigment, how much of that pigment is produced. These genes follow the incomplete dominance pattern, and each dominant allele contributes a unit of darkness. And it's a cumulative effect. So if you have 2 dominant alleles, your skin color will be twice as dark as if you only have 1 dominant allele. If you have 4 dominant alleles, then your skin tone is going to be 4 times darker than a person who only has 1 dominant allele. And so I think you'll understand that better when we look at an example. Okay, so again, this is a way oversimplification because this model that I'm sharing with you assumes that we only have 3 different genes contributing to skin pigmentation. It's definitely more than 3, but this is kind of a simplistic way to conceptualize it. So notice a person who is homozygous dominant for all 3 genes is going to have very, very, very dark pigmentation. A person who's homozygous recessive is going to be on the other end of the spectrum and have really, really light pigmentation. So just imagine somebody from Northern Europe who looks almost like a ghost. Okay, and that would be the other end of the spectrum. And then most people on the earth are somewhere in between, right? So a person who has a mixture of dominant and recessive alleles is going to have a medium level of pigmentation. But notice also, it doesn't matter which alleles are dominant and which are recessive. So this person here has 1, 2, 3 dominant alleles and 3 recessive alleles. This person also has 3 of each, but it's not the same exact pattern, and yet the skin pigmentation level would be the same for these 2 individuals. So it's the total number of dominant alleles and not necessarily which specific dominant alleles. And so it's what we call a cumulative effect or an additive effect. So here you have a marriage between 2 people, both of whom have that medium, moderate amount of skin pigmentation. Here they're using the dark circles to represent the dominant alleles, in this case big A, big B, big C, and then the white circles represent the recessive alleles, little a, little b, little c. And if you do, this would be a tri-hybrid cross. So notice we have 1, 2, 3, 4, 5, 6, 7, 8 different kinds of eggs and 8 different kinds of sperm, and therefore 64 possible offspring. Notice, the main thing to notice here is the different shades of pigmentation. It's not just dark and light. It's not just dark, light, and medium. There's a whole spectrum possible. So even in this simplistic, way oversimplified example, there's 1, 2, 3, 4, 5, 6, 7 different levels of pigmentation that are possible in these offspring. And again, the greater the number of dominant alleles, the darker the skin, and the greater the number of recessive alleles, the paler and the less pigmented the skin is. So that's polygenic inheritance. Poly meaning many, genic meaning genes. So this is many genes contributing to one trait. Many genes, one trait. Polygenic inheritance. Okay, it's also worth noting that many characteristics, and certainly many human traits, are determined only partly by genetics. It's the whole nature versus nurture debate. And it is true that both nature and nurture, in other words, both our genes that we inherit from our parents and the things that happen to us during life, both of those can have some effect on our phenotype. So for example, human skin pigmentation, as you just saw, is determined to a large extent by the genes that were passed down to you from your parents. But we all know that the more time you spend in the sun, the darker, at least temporarily, the more pigment your skin will produce. And so you spend a lot of time exposed to ultraviolet radiation, you're going to get a tan. Okay. Now, do some people have a greater capacity for tanning than others? Absolutely. That's the nature part. That's the genetic component. But all of us, or at least most of us, have some effect on our pigmentation just from being in the sun. Human musculature, how muscly you are, is of course partly determined by the genes that were passed down to you from your parents. But no matter what your genetic predisposition is, if you go to the gym frequently and you work out and you do it in the right way, you're going to build extra muscle. You're going to become more muscular. So again, that's partly genetic, partly affected by environment and experience. Human intelligence definitely has a genetic component. There's no doubt that if both of your parents have a high IQ, if both parents have a high intelligence level, you are more likely, now there's no guarantees, but you're more likely to be intelligent yourself. However, we also know that human intelligence is greatly impacted by experience, especially early life experience, things like parents reading to their children, parents communicating, talking, stimulating their children by interacting with them. That helps the brain develop. And so intelligence is partly determined by your genes. It is also greatly affected by the learning experiences that you have early in life. And finally, even diseases like cancer certainly can have a genetic component. There are some genes that we can very specifically link, some types of cancers that we can very specifically link to particular genes. A famous example of that is a gene called the BRCA1 gene. It's a mutated allele that can be passed down from parent to child. And anyone who inherits a copy of this allele, a woman who inherits a copy of this allele, has a 70% lifetime risk of developing breast cancer. Now, your average woman only has about a 12% lifetime risk of breast cancer. But if you inherit this gene, your chance goes from 12% to 70%. So that's a big jump. And so obviously this gene contributes to the likelihood of developing that cancer. But then there are other cancers that really are not very much linked to genetics at all, but rather are caused by environmental exposure to what we call mutagens, chemicals that alter our DNA. So cigarette smoke, for example. And many times cancer is both. You know, there are certain genes that we inherit that maybe give us a greater predisposition to cancer, make it more likely to develop a certain type of cancer. But then exposure to things in the environment, in the foods that we eat, in the the things that we're exposed to in our daily lives, those things also can either increase or decrease our risk of certain types of cancers. Okay, I'm looking at the clock. I'm already running at almost 40 minutes here, but I'm going to keep going because I've only have one more topic to discuss in our non-Mendelian genetics lesson. And so I want to talk about what's called X-linked or sex-linked inheritance. And before we get into that, it's important to note how sex is determined in humans and in fact all mammals, and that is by what we call the sex chromosomes. So you know that the somatic cells of a man, for our species, for humans, the somatic cells of a male have 46 chromosomes. That's 44 autosomes and 2 sex chromosomes, which would be an X chromosome and a Y chromosome. In women, human females, likewise, there's 46 chromosomes total. 44 of those are, again, what we call autosomes. That's regular chromosomes that have nothing to do with being a boy or a girl. And then there's one pair of X chromosomes, which is what makes females female. XY makes the child develop into a male. XX will develop into a female. So when gametes are made, when meiosis occurs in the ovaries to make eggs, every single egg should get 22 autosomes, that's half of the autosomes, and one copy of the X chromosome. But in men, when sperm cells are made, half of the sperm are going to get the X chromosome and the other half of the sperm are going to get the Y. So what does this mean? It means that sex is determined by sperm and not by egg. because the egg always carries an X chromosome, 22 autosomes, one copy of X. That's what happens in the egg. But the sperm carries either an X or a Y. So if this sperm fertilizes this egg, it's going to be a girl. But if this sperm fertilizes the egg, it's going to be a boy. Okay? So XY for boy, XX for girl. That's how sex is determined. Now, it turns out that while the Y chromosome pretty much only has one purpose, and that is to make boys boys, the X chromosome carries lots of genes that are needed by both boys and girls, which is a good thing because notice boys and girls both have at least one copy of the X chromosome. So any trait, any inherited trait that is determined by a gene carried on the X chromosome, that trait is said to be X-linked, or we call it sex-linked. Okay, so examples are hemophilia, which is an inherited disorder where the person is missing a particular protein in the blood, so the blood does not clot properly. A small injury will just bleed and bleed and bleed, and it really doesn't stop without medical attention. Duchenne's muscular dystrophy is where there's a dysfunctional muscle maintenance protein that leads to progressive weakening of the muscles. It affects walking, and eventually it results in a fatal loss of the ability to breathe. Adrenoleukodystrophy, ALD, causes gradual loss of myelin on the nerves, especially the motor nerves that allow the brain to communicate with the muscles. So these young men eventually lose their ability to walk and talk, and eventually the nervous system shuts down, and it is a fatal disease. And finally, red-green color blindness, where a person can see color, but they cannot distinguish between the two colors of red and green. All of these are inherited traits. The genes are carried on the X chromosome, and for all of these sex-linked traits, they are much, much more common in men than in women. So let's see if we can understand why. First, I'll give you a simple test. I want you to look at your screen, look carefully at the dots. See if you can identify a two-digit number. And think to yourself what number you see. What about these dots? The second circle. Can you see a number? Perhaps a two-digit number? Look at it carefully. It's a little harder this time. Okay, so the first test was the control test. Everybody should have seen a 12. Okay, the second one is the experimental test, and what number you see depends upon whether or not you have normal color vision. If you see a 74, then you probably have normal vision. If instead you see a 71, you probably have red-green color blindness. Now keep in mind, this is not a completely accurate test because your screen may not be properly calibrated for these colors, and so that may affect how you did on your test. Let's use hemophilia as another example of a sex-linked trait. Because this gene is carried on the X chromosome, we're going to write it out this way. So X big H, that would be a normal female whose blood clots completely normally. Heterozygous, X big H, X little h. This is a woman who has a normal blood clotting allele and a hemophilia recessive allele. So her blood's going to clot just fine. She's not going to have symptoms of hemophilia, but she's a carrier, and she can pass this down to her children. And then here's why it's extremely rare for a woman to have a sex-linked trait, including hemophilia. Because for a woman to have hemophilia, both of her X chromosomes have to have this recessive allele. That means her dad would have to have the disease, and her mom would have to at least be a carrier. And that's just not going to happen very often. That would be really, really unlikely. But it could happen. We do know from history that Queen Victoria, who was on the throne in England back in the 1800s, and she reigned for many, many, many years, she was definitely a carrier of hemophilia. I can show you the family tree, and I'll show you that in a moment. For men, because we only have one copy of the X chromosome, and these genes are only carried on the X, it's much more common for a man to have hemophilia, because a man would only have to inherit one copy of little h. See, there's nothing on the Y to cover it up. So a boy is either X little hy or X big hy. There's no in between. There's no such thing as a carrier male for sex-linked traits like hemophilia. Okay, so I mentioned Queen Victoria of England. We know she was a carrier, and if you notice, Victoria and Alice and let's see, Alfred and Helena and Louise and Arthur, Leopold, these were all, and Beatrice, those were all her children. Her son Leopold inherited that hemophilia allele, and this was kind of unusual because he had hemophilia and lived long enough to marry and produce kids. Of course, the downside of that is he was able to pass that hemophilia allele. You can see it continued to go down through the generations. In the 1800s, most boys born with hemophilia did not live long enough to marry and reproduce, so he was kind of unusual for his time. But again, this is just an example of how traits like this can pass from one generation to the next. And also, just in case you're curious about how Queen Victoria's legacy connects with the modern royal family, here's where we are today. This is Queen Elizabeth, who currently reigns in England. Her husband, Prince Philip. This is interesting that their family trees are connected. Elizabeth and her husband are third cousins, which is kind of interesting, but not that uncommon in the royal family. Prince Charles is the future king of England here. But notice they have dodged a bullet because this whole wing of the family does not have the hemophilia allele. But notice the other parts of this family certainly do have it, all passed down from Queen Victoria, who was a carrier of hemophilia. So let's work a problem. Abe has hemophilia. Obviously, he's a male. He has hemophilia. He marries Bonnie, who is a carrier. So we're going to show this on a Punnett square. Abe has hemophilia, so X little h, and he's a male, so we're going to give him a Y chromosome. You do not put a gene on the Y, because remember, sex-linked traits are only carried on the X chromosome. And he marries Bonnie, who is a carrier. This is what makes her a carrier. So she does not have hemophilia, but she is a carrier. And if we look at all of their possible children, of course, we would expect there to be an equal probability of girl versus boy. And you can also get an idea of how likely it is that they would produce a child with hemophilia versus a child whose blood clots normally. So what is the probability that Abe and Bonnie's firstborn child will be a hemophiliac son? Well, here's a boy. This is a normal boy without hemophilia. Here's a son with hemophilia. So that's one possible child out of 4 possible children that they could produce. So the probability that their firstborn child will be a hemophiliac son is 1 out of 4. So it's the probability of having a son, instead of a daughter, which is 1 out of 2 chance, multiplied times the probability of the child having hemophilia, which is also a 1 out of 2 chance. So if you multiply, rule of multiplication, you multiply those two 1/2, you get 1/4. Hemophiliac son is a 1 out of 4 probability. Now, separate question, totally different question. Same Punnett square, but this time listen to how I worded the question. Bonnie gives birth to their firstborn child. It is a boy. Now, what is his probability of having hemophilia? The difference is the way this is worded, the kid has already been born. We know it's a boy. So do we really need to consider these 2 possibilities? No, because these are girls. If we know it's a boy, he's either this or this. So his probability of having hemophilia now is a 1 out of 2 chance. That exact same Punnett square gave us a 1 out of 4 probability before. The difference is the way the problem was worded. See, this is future tense. What is the probability that their firstborn child will be? Kid hasn't been born yet. So we don't even know if it's a boy, might be a girl. But the probability that that child will be a hemophiliac son is 1 possibility out of 4 possible kids that they could have. But if the kid has already been born and we know it's a boy, then it's a 1 out of 2 probability that he will have hemophilia. Okay, and I always like to leave you with one to do on your own, so please try this out. Mike is a boy who suffers from hemophilia. Both of his parents have blood that clots normally, so neither his mom nor his dad has hemophilia. Use a Punnett square to show the cross between Mike's parents, and then you'll be able to answer this question: from which of his parents did Mike inherit this disorder? So read that again quietly to yourself, pause the video, and work out the Punnett square. Okay, now, first of all, Mike is a boy, right? So I know he's going to have an X chromosome and a Y chromosome. He's a boy with hemophilia, so he has to have a little h on his X chromosome. So this is Mike, okay? Obviously, he got the Y chromosome from his dad, okay? Because otherwise he would be a girl, right? If he got the X chromosome from his dad, he'd be a girl. So he had to get the Y chromosome from his dad. That means that little h that Mike got was inherited from his mom. The problem states that neither his mother nor his father has hemophilia. So I knew I had to put a big H right here for dad, and I also know that mom has to have a big H because it says both of his parents have blood that clots normally, right? But I also know that mom has to be a carrier. That's the only way Mike could have inherited that little h. So from which parent did Mike inherit hemophilia? And we can see clearly that would be the mom. All right, and this is a good place to stop. This is the end of our lesson on non-Mendelian genetics. I hope you've enjoyed it, and I hope you've learned at least a little something. And I'll talk to you soon. Take care.